Hello students, so today we will start the third chapter of the class 8 biology book of Telangana state, and as you can see the chapter is on the world of microorganisms. Now you must have remembered that chapter 2 was has the same name, but it was part 1, and in that part we effectively talked about many different things regarding what are the various groups of microorganisms. I have shown you live videos of microbes in action. Okay, we have talked about it. Now let's see about a different aspect in this chapter, and that is how they are useful to us, and again what are the ways in which they can be harmful to us. Okay, we have to study this and understand them. Then it will be better for us. We will be able to use them in a way. Suppose you know that somebody is going to hurt you, then you have to take precautions, isn't it? So to start with, you have to understand what are the things which affect the microbial growth. Okay, let's first start off with it. Now you see, what is actually happening is that microbes, they are just multiplying. Remember, I told you last time, 1 becomes 2, then 2 becomes 4, and similarly each of them will keep on multiplying. Okay, this is the thing. They multiply much faster than us human beings. Okay, now so this is all the story of their population. They keep on growing at a very fast rate. Their population increases at a very fast rate, and we have to understand what are the factors. Well, you see, the first is availability of nutrients. Of course, if they have got the right nutrients, then their growth will be much faster. That means this process of 1 becoming 8, it might take 1 second in one case, and it might take even 10 seconds depending on all these factors. The first is availability of nutrients. Next is optimal temperature. You know, my dear students, for the microorganism, each of them, you name any bacteria, okay, any protozoa, any fungi, more or less there exists something like this, a dependence on temperature. This temperature dependence is somewhat like this, something like this, as you can see. And what is the meaning of this curve? Let me tell you. So on this axis we have got the temperature, and on this axis we have got the population, their numbers. So what you see over here is that there is an optimal temperature. This is the optimal temperature where the population is very high. So this is very good for them, the population. This thing will happen much faster. Say in this optimal temperature zone, this 1 to 8, just from 1 to 8, this thing will probably happen in 0.001 seconds. But in other places over here, you see, as we decrease or as we increase the temperature, here the conditions are not fruitful for them, and so here the same thing will happen, but it will take much longer. Probably it will take 2 seconds or it will take 3 seconds. So you have to understand that for the microbes, they have this optimal temperature where their population is maximum. Now if we look at a different microbe, it might have a different structure like this, temperature dependence. But one thing is important, this peak does happen for each and every microbe, this dependence on temperature, and we have to understand this feature. Suppose there is a microbe we want to take care of, that is, we don't want it to multiply. Okay, so what do? We keep the temperature either very high or either very low, then the population will decrease. Remember, we cannot entirely stop their population. For that you have to decrease or increase the temperature much high or much low, but that is often not very possible. We use refrigerator for lowering the temperature. You remember when you get milk, okay, from the milkman, you boil it. That is effectively increasing the temperature. See, the same old principles working here. Okay, now what's the other factor on which their growth depends? Well, moisture, that is presence of water. Of course, we all drink water. If we don't drink water sufficiently every day, our health will be affected. And so, this is the same thing for the microbes. Come on, they are living organisms. Next, presence of oxygen. Okay, the same thing will happen. Some of them use aerobic respiration. For them, oxygen is important. Others use anaerobic respiration. It will not be that important. Again, presence of light. Okay, so these are some of the factors, and you will learn about these things more in your higher classes. So this is just an extra information I thought you, my dear students, must have if you want to properly understand the inner workings of microorganisms. Okay, so let's start with our chapter. Now, as I have already told you, we have seen that in fact these microorganisms are omnipresent. That means they are almost everywhere. They are present in air, water, soil, well, even within our bodies. Interesting, isn't it? Last time I told you that they help us greatly in digestion. Okay, so as I was saying, some of them are useful and some of them are very harmful. So we have to take precautions, and for useful ones we have to use them for our purposes in a better way so that our life can be better. Okay, let's start then. First we will talk about our useful friends. Okay, useful microorganisms. Well, they are useful to us in several ways. Let's start. Say for example, in making of curd and in preparation of idli, dosa, bread and cake. I love each and every one of them, and so I thank the microorganisms for helping us silently. Come on, if you go to a restaurant or anywhere, you have to pay to eat this, but we don't pay these microorganisms anything because it is their natural work. So what is their natural work? That we will learn now. You see, some microorganisms are again useful in preparation of medicines required to cure different diseases. So can you imagine what are so these are some very interesting applications, my dear students. Now, some of them are farmer friendly as they increase the soil fertility. Okay. Now, remember, soil fertility means if soil is fertile, more and more crops will grow, right? We have a huge population in India. So soil fertility is very important. So you understand the great role played by our great useful friends, good old friends. I must write it over here. So these are our good old friends. Okay. Now, let us take some lukewarm milk in 2 bowls. Add a few drops of buttermilk or little curd in one of the bowl, and in the second bowl you don't add anything. This thing you can do at home. Okay. Now keep the 2 bowls undisturbed in a warm place and observe the milk in the bowl after 5 to 6 hours. Okay. What changes do you observe? You will observe that the bowl where you have added the small amount of curd or buttermilk, it has entirely converted into curd. Now, what is the reason for this? You see, the reason is simple. So we have the two bowls, right, with milk in it. Now what has happened is that when the two bowls, say bowl A and bowl B, when you introduced some of the curd in it, then you see these curds were not what was there in the curd. You have to understand scientifically, there were small bacteria in it. These are known as Lactobacillus. Remember last time? See, this is the page 30 where we introduced the microorganisms, section 3.2. Remember, here I showed you Lactobacillus. Just look at them. So if you look into the milk with a microscope, you will see something like this. Look at the structure. Okay, how do they look like? So this is a bacteria, my dear friends. And what do they do effectively? What is there in a small amount of curd that we put in the bowl? Okay, now what happens is that some of the lactobacillus we introduced into the milk, right? Now what is their work? If we put them in a certain warm condition without disturbing them, no light, no sound, nothing, then they will keep on multiplying. This one becomes two, then what will happen? And this thing will happen very quickly. So this one lactobacillus, suppose this is one lactobacillus, this becomes two. Okay, then two will become, the same thing will happen, two will become four, and so on. So see, so this thing will happen. So after 5 to 6 hours, what has happened is that the milk is the same, but what has happened is now there are much more lactobacillus bacteria in it, and their presence has actually some other effects as well. So let's learn about it. So effectively we are talking about changing milk into yogurt or curd, right? Now what happens is that lactobacillus bacteria feed on the sugar in the milk and they keep on multiplying. Remember the factors I told you about? I was just talking to you, right? That they multiply depending on several factors. Now here what has happened, we have given the lactobacillus availability of nutrient, no light, no sound, perfect conditions. We have given lots of available nutrients in the milk. Okay, so what they feed on is the sugar in the milk, and they change the sugar into a weak acid called lactic acid. Okay, and during this conversion we are just getting the curd as a result. Remember, the sharp taste of the yogurt is due to the lactic acid. Now think of bread, yet another example you will find in the text. Okay, think of the making of bread. You see, take 100 gram of maida in a bowl, add 1 or 2 spoons of yeast powder, add some hot water, knead it into a dough. Now keep the dough in a warm place. Okay, now observe the dough after 3 to 4 hours. Remember, in both of these activity we are keeping them undisturbed for few hours. This is important for bacterial or whatever microbial population increase. Okay, you have to keep them undisturbed. Now what changes do you observe? See, look at yourself. Maida dough, see, the size has increased. That is what? How did it change? Have you ever wondered? You might have observed your mother preparing fermented food like idli and dosa. So what could be the reason for preparing idli and dosa dough a day before cooking it? Okay, what do you think is the reason? Well, you see, these are the 2 cups, right? In bakery, when yeast is added to the dough for preparing bread, the dough rises. So which in out of them, in which cup do you think the yeast was added? Of course this one, because you see the dough has risen, right? Now this is due to the production of carbon dioxide gas during this process of fermentation. and what is happening is that the bubbles of gas, they keep on going out, and they go out. This makes the duff spongy in nature. So think of it, there is a material, bubbles of carbon dioxide keep on going outside, and so the whole material is increasing. Okay, this causes an expansion of the material, the duff material. So this is what is happening. Now you might be wondering, where does this carbon dioxide come from during this whatever called fermentation process? In simple words, what is happening is that the yeast, when you add it to the bread or duff, it feeds on the nutrients on the flour. Okay, just similar to the previous case. The bacteria were feeding on milk, on sugar in the milk. Okay, now this time they are feeding on the nutrients in the flour, and when they respire, it makes carbon dioxide gas. So the thing which increases the duff, okay, as a result of which the duff rises, it is due to the production of carbon dioxide by these yields due to their respiration. And the more their population increases, the more they respire, and as a result the duff increases much faster. Okay, so that's the whole idea. Okay, now let us look at yet another activity where we will be considering again commercial use of microorganisms. Now again let us take two bowls, okay, half filled with water. Now add 5 to 10 spoons of sugar in each of them, and then add 2 to 3 spoons of yeast to any one of the bowls. Now close both the bowls with lids and keep them in a warm place after 3 to 4 hours. Okay, now this time we are just doing the activity with water, okay, and plus yeast. Okay, now what differences did you observe between the two bowls? Okay, you will see that there is a characteristic smell of alcohol in the bowl where you have added the yeast. But why is this happening? Actually, what have you added in both of them? You have added sugar plus water, right? And then you have given yeast. So what is happening? The yeast is feeding on effectively both sugar and water, effectively sugar, right? And this is the reason, you see, sugars are converted into alcohol by yeast, and this process of conversion of sugar into alcohol is known as fermentation. This process is used on a large scale in the production of alcohol, wine, beer, etc. And for this, yeast is grown in natural sugars present in grains like barley, wheat, rice, and crushed fruit juices like grapes. So let's look into a little more details. Well, fermentation is effectively, as you can see, it is a metabolic process where these microorganisms like yeast and bacteria, they break down carbohydrates like sugars into simpler products. Okay, so we have sugar and it is broken into simpler products, and this breaking down is done in the presence of these microbes. Okay, they are the ones, they are the soldiers who are doing this work for us. Okay, and this entire process is fermentation, and remember, there is no oxygen required for this process. Okay, and this process is used to produce various foods, beverages. See, for example, we have sugar plus yeast, no oxygen we don't require, and you see the yeast works on the sugar and it is broken down into carbon dioxide and alcohol. We just don't want this carbon dioxide as well, and we just take our alcohol. So that's the basic idea. Okay, before I proceed further, my dear students, let me again give you a microscopic view of what is happening during fermentation, and I have taken the help of YouTube to show you this. Okay, so this is the video of this channel, Science Spy. So let me show you what is happening. See, wow. So under favorable conditions, you see what is happening. You understand what is it you are seeing? This is just the fermentation process. Look everywhere. You see? So they are adding active dry yeast. Okay, see what is happening. Then sugar, then water. Okay, and then the dropper is used to mix all of them, and see the result. This is a time, how much time has elapsed. You can see over here 20 minutes has elapsed. Look what is happening. Slowly the yeasts are getting their food. They were so bored in the plastic packet, but now let us observe what is happening in the microscope. See the slides and see this is happening. Wow. So see, this is where their population is increasing, and see this is probably the bubbles rising. Okay, they are getting converting. Okay, see, wow, just after 3 minutes, see this is the entire structure. The population is keep on increasing at a much faster rate. So this is effectively yeast fermentation, my dear students. It's an amazing microscopic world when you see it with microscope. Unfortunately, we can't afford to have this with us, so we are taking the help of people who have done such activities. Okay, so let's proceed then. Okay, so now let us talk about the medicinal uses of microorganisms. Now, of course, you must have experienced falling ill yourself, right? It's a natural process of growth in a human body. In fact, doctors say that it's good that you fall ill maybe once in a year. Why? Because this will mean that the human body is working perfectly. When you fall ill, there are various defense mechanisms into your body. They come into play and they try to help you recover from the foreign infections. Falling ill is basically due to something. Remember our cell, so think of your body like a cell. Some foreign particle is entering your body and which is disrupting the normal mechanism of your body. So that's the basic idea. Now our body has its own defense mechanisms, own microbes as well as defense cells and all that. Not just microbes, effectively we have got defense cells. Okay, RBC, WBC in general in the blood, and there are others as well. I'm not talking about the details. Now whenever some foreign particle enter, they attack and fight with it. Okay, so that's the basic idea. Now, but you also know that sometimes these things don't work. The mechanism is not sufficient to treat the foreign infection. Okay, the foreign particles invading mechanism. So we need external help. And what is the external help? So suppose, think of it, think of the foreign invaders, in this case as bacteria. Okay, now when there are so many bacteria, so this is a human body, think of it like that, and then we have got some bacteria from food or some other sources. There was a cut in your body. Okay, you were playing around and you got cut, and the result of it, there was a cut in your body, and from there you get bacterial infection. What is happening? Bacteria entered your body and now they are quickly multiplying. What to do? You go to doctor. Okay, you get TEDback and whatever. Sometimes even if you develop infections into your body, not just cut, it's something different, you develop infections, then this is due to the growth activity of the bacteria. Now in this case, what you take are these medicines known as antibiotics. Now what are they? Effectively, the doctor prescribes you antibiotic tablet, right? inside the tablet are other microbes. Don't worry, they are your useful microbes. That's why I am showing them in green. They are good. Now when you eat them, you inject them, this tablet gets broken down into smaller parts and the microbes are released. Okay, then these microbes, they enter the target site where the bacteria is actually trying to invade in your body. Okay, now these microbes, which are basically antibiotics, they make life difficult for this bacteria infections. And that's the basic idea of working of microbes. How do you think this happens? Now here I have written it over here, the overall summary. Effectively, we are talking about bacteria, right? What's that? A single cell. Think of it. Now we have to affect the cell's function. We have to affect. And how do we do that? Effectively, these microbes, which are part of antibiotics, they disrupt the essential functions within the bacterial cell, either killing them or stopping their growth. Now, what do? They effectively target various aspects of bacterial life. And how do they do that? They do that by interfering with essential bacterial processes, that is essential processes which happen inside the cell, like cell wall synthesis, okay, next protein production, nucleic acid synthesis, all the things which happen inside the cell, which are required for what? For the sustenance of the cell, so that it remains alive. Now, as a result of this disruption, what happens is that either the microbe just dies or it is unable to multiply, and this is the way of treating bacterial infections. So the basic idea is bacteria, they are multiplying, right? So the basic idea is to stop the multiplication. So for that, either we kill the bacteria or we stop this entire growth. Whatever has happened has happened. Don't overdo your multiplication any further. That's the basic idea, my dear students. Now, in this channel of ours, YouTube channel, whatever video you see, you will find that I always try to give you something extra. This may be of some other higher class, okay, it doesn't matter. What I want you is to understand the basic mechanism, basic idea of how our world is working. Okay, you have to be curious enough, then only you will become a good student who is able to understand the things in a better way. So now let's continue. So as I was saying, doctors prescribed antibiotics, and these are produced by growing specific microorganisms, for example fungi. So this bacteria, the invaders that we have, and we sent our soldiers, and the soldiers were small fungi soldiers. So think of it as a war between bacteria and fungus. Okay, interesting concept, isn't it? Yes. Now, nowadays, a number of antibiotics like penicillin, tetracycline, streptomycin are there. Okay, they work on different types of bacteria. Okay, again, target sites probably are different. Now, antibiotics help in curing many bacterial diseases. Let's name few: tuberculosis, gonorrhea, diarrhea, typhoid, and prevent infections like septicemia. Now, you see, not just humans, they are also used to contain bacterial diseases in plants and other animals as well. Look, these are antibiotic injections. Vial is there, you put the injection inside, take the fungus outside, inject it into human or plant, no, human or any other animal. Okay, and these are again another way of taking antibiotics, antibiotic capsule, which I've just shown you previously. Okay, before moving forward, let me again have some more discussion with you. So remember, last time I was telling you, suppose you are playing around and you got cut somewhere. Now you see what is happening is that from the cut some bacteria invades your body. Say for example, and to prevent that, and that bacteria is effectively very dangerous, and we have to take tetanus shots to avoid that, to prevent tetanus, okay, which is a serious fatal infection that can be fatal. Okay, very dangerous, students. You play around, you get a cut, especially with rusted iron or something, then on the iron rust over there is this bacteria, and which can cause tetanus, which is also known as lockjaw. Now what happens is that this bacteria release a toxin that causes muscles to tighten and stiffen. Okay, now vaccination effectively helps the body develop immunity against this toxin. And so what is vaccination? Well, that's the next topic that we will soon cover. But before that, let me show you the live effect of an antibiotic on microbes. You will see what is happening. So this is the exploding bacteria with penicillin, another video. On this side you can see the effect of penicillin. See the bacteria, they are exploding, but on this side they are healthy, they are moving around. See the bacteria, they are exploding. Why are they exploding? You must be wondering. Again, let me show you. See, so this one is exploded, this one is exploding, this one is exploding, this one is exploding. Why? What is happening is that in this case particularly, so if we have a bacterial cell like this, so what is happening, the penicillin has caused inhibition of the cell wall. Cell wall will not develop further, and that's why what is happening is that there is crack or somewhat in this, and the material blows out from inside the cell, because the cell wall is now become sort of, sorry, not cell wall, cell membrane, it has now become somewhat unstable, and so the bacteria, the material explodes, and this is causing the cell death that you have just witnessed. Now let me tell you something very interesting. So what I have told you till now is that if this is a human body and we have foreign invaders that we can't handle, then we take in antibiotics. Foreign invaders must be of bacterial in character, so you have to visit doctor to identify whether it's a bacteria or some other microbe. Now the next thing is, so we can take antibacterial medicines without even prescribed by the doctor, is what you might be thinking, to save money, doctor's fees. Well, you see, you should not do that. The first thing, identification of the microbe. Next, if we unnecessarily use antibiotics, so suppose whatever has invaded the body of a human being, they have been killed by the antibacteria, fungus. Okay, but now you are still taking on medicine, that is, you are still taking on injecting more and more fungus which are inside the antibacterial medicine, which are the antibiotics itself inside your body. What will it affect? What happens is that if you take lots and lots of antibiotics, they will affect your blood cells. These blood cells are our vital soldiers, defenders. They are inner body defense mechanisms. Okay, now they will affect the working of the blood cells, and so what will happen is that next time if there is some infection, these blood cells will not respond as they should, their working will be affected. Okay, and what is the meaning of this? The meaning of this is that they affect the blood cells which fight infection, and also they cause resistance towards antibiotics. So if we keep on taking more and more antibiotics, soon we will develop a resistance towards it. It will not be good. So if you are young, and you are taking lots of antibiotic, then when you get old and you get some bacterial infection and you take antibiotics at that time, it will not help you at all. Why? Because too much excessive use of antibiotics has caused your body to develop a resistance towards it. So as you age on, you will face great problems. Okay. And another thing, so there are three important effects of unnecessary use of bacteria. Number one, they affect blood cells which fight infections. Number two, they cause resistance towards antibiotics. And number three, they kill useful bacteria in our body which are there to help us in digestion, in several other processes as well. So this is very dangerous. Now let me tell you about something very important. You must have known that nowadays chicken that we eat, okay, that is made in large numbers in farms, okay, chickens are produced in large numbers, they are grown much faster by the use of antibiotics. Okay, now if we eat chicken a lot, what happens is that the antibiotics which is still present inside the chicken gets inside our body. So effectively, by eating lots and lots of such type of chicken which have got lots of antibiotic in it, we are effectively eating lots and lots of antibiotic tablets, and soon we will again develop resistance towards other antibiotics. Even our body functions will be affected by both the actions on the blood cells as well as killing of useful bacteria in our body. Okay, so you have to be very careful nowadays. Next, let us little bit learn about the story of penicillin, the antibiotic. So how was it discovered? How did we know that effectively what we are having? Think of it as a situation. We have a fort. Our body is like a fort where some foreign invaders have attacked bacteria. They are the foreign invaders. Their army is attacking us. Okay, so that's the situation. Now we have called in hired mercenary. We have paid money to some soldiers. They are hired soldiers, so they are the mercenary. This is the fungi, okay, bacteria. We are using tablets. We call them, they come and fight with this bacteria, but they have already invaded our body. So the hired mercenaries also have to enter our body, and the fight begins inside our body. And how do you know the fight is happening? You get fever, your body temperature rises. Why? Because the blood has to flow a lot. Blood has to flow in, taking these antibiotics to different sites where bacteria has invaded, and that's why due to the high rate of flow of blood, your body temperature rises. So these are some basic ideas about basic human biology. So effectively I am giving you a sort of crash course on human biology. Okay, so you can become a junior doctor yourself, sort of. You don't have to take any degree or whatever. You have to try to understand how a doctor thinks and works, how a biologist works. Okay, effectively you are doing the work of a microbiologist at the time. Right now you are trying to understand how these microbes are working, their inner biology. Okay, good. Now you see, this great person is Dr. Alexander Fleming. So he is the one responsible for the discovery of penicillin. Well, he was an army doctor during World War, and he observed that many injured soldiers died because of bacterial infections of wound, right? And what was the infection, do you think? It is just this culprit, Tedback-like infections that we have just talked about. Okay, so it was very dangerous at that time. Nowadays also is dangerous, but we at least know how to defend ourselves against it. Okay, so now he was working on antibiotics in his lab. As part of his research, he grew groups of bacteria in petri dish, Accidentally, one day he observed that some fungi were preventing the growth of bacteria. Okay. In the petri dish. Petri dish is small bowls and like stuff. Okay. Now, in which he was growing bacterial colonies. So he separated the substances released by the fungus and tested it on some other disease-causing bacteria. He came to know that this substance also prevented those diseases causing bacteria, and the fungus was identified as Penicillium notatum. The substance which was produced by the fungus is penicillium, which was later named as penicillin. Okay, so you see the discovery of antibiotics, penicillin, was announced in 1929. By 1945, Dr. Alexander Fleming was awarded the Nobel Prize along with other scientists, namely Dr. Howard Florey and Dr. Ernest Chain. The discovery of penicillin paved the way to the discovery of many antibiotics like streptomycin, erythromycin. Okay, good.